Hello, BISC 210 students. Welcome to Fall 2020 semester. This is Chapter 1 video lecture. This is the first one in the semester. I will be going over some really basic biology as well as microbiology in this chapter. It's possible it could be a little bit of a review for some of you, but for some of you, information may be new. Please pay attention carefully, take notes, etc. So, in this chapter, we're going to focus on several topics, including this first one, which is basically what are microbes and how do we define them and where do we find them, as well as how do they affect our lives, because there are more bacteria on your hand than there are people on the planet. So they're everywhere and they play a really important role in a lot of functions. We have different types of relationships with bacteria and other microbes. So a microorganism is, by its true definition, it's just anything you really can't see with the naked eye. You should be able to hopefully name a few things from that. Give you a second or so to try and think that over. Some organisms that can't be seen with the naked eye. Okay. Well, you could include in that list bacteria, viruses, archaea, protists or protista, even fungi. Now, I'm not talking about the fungi that grow in the woods that, we can forage and eat. We're talking more about molds and their spores. So, for instance, molds are a type of fungi, and one of the more famous ones is aspergillus, which causes aspergillosis. But it's a mold that releases the spores, and when you breathe in the spores, they go to your lungs and colonize and wreak havoc on your lungs. So, bacteria, viruses, archaea, protists, and some fungi, specifically molds and yeasts. Alright, so a lot of people use the term germ pretty often. They mostly associate it with being sick. Technically, this is a incorrect use of the word. Germ is just replying to a rapidly growing cell. So let's say you have a specific bacteria that grows in your gut, your GI system, and you take an antibiotic. The antibiotic will kill a lot of those bacteria in your gut, and we have to reestablish those organisms, and the faster they grow, the better. So we could refer to them as germ cells. But a lot of people tend to confuse that with being sick. Yes, you could technically, on a technicality, you could say, because I am sick, I have germs. Meaning you're sick, you have a disease-causing microbe that's growing fast. It is possible, but it's not the best use of the term. Now, a few microorganisms are pathogenic. Pathogenic means disease-causing. So there are most bacteria are not disease-causing. About 2% of the known species of bacteria are pathogenic. Usually less than 2, actually. Depends upon what book you consult. Some books say around 1% to 1.5%, and then up to 2%. So anything below 1% or anything below 2% is a safe answer. So a pathogen is something that is a microbe that can cause disease. So, for instance, coronavirus is a pathogen. It causes disease. That's an example of a viral pathogen. Bacterial pathogen, chlamydia, gonorrhea, syphilis. They're bacteria, but they still cause disease. They are pathogenic. But again, pathogens are such a small percentage of the known microbes on the planet. Most of them are non-pathogenic. And that's a good thing, because obviously if there's a ton of pathogens, we would be at risk all the time. In our current predicament, we are kind of at risk all the time. But in a normal pre-COVID world, we really weren't significantly at risk for a disease unless there was an outbreak and or you're someone who has a compromised immune system or underlying medical conditions. All right, so let's move on from that. So types of microorganisms, prokaryotic versus eukaryotic. Oops. You can see here, I kind of listed most of these earlier, but I just want to reiterate that bacteria, archaea, fungi, protozoa, even some algae, viruses, and multicellular animal parasites. Now, that one is interesting because when you say multicellular animal parasites, you're talking about things like a tapeworm or a flatworm. These organisms can colonize your body and steal nutrients from you. But if you think back to what we defined as a microorganism as something we can't see, it doesn't mean we can't see it forever. It means that it spends most of the time being able to not be seen. In the case of tapeworm, it's larval stage, not the adult stage that we can see. The larval stage, when it's in its egg and developing, we can't see without a microscope. It's too small. So as long as part of their life cycle is in the falls under that umbrella of not being visible with the naked eye, then you can still consider it a microorganism. Now, at the top, there's in parentheses 2 terms, prokaryotes and eukaryotes. I'm hoping that I don't have to spend too much time going over basic biology. Hopefully you've had at least some form of biology at some point. Prokaryotic versus eukaryotic organisms. There are some major differences. Eukaryotes tend to be unicellular. So prokaryotes are unicellular. And really the big thing is they don't have a nucleus. So bacteria have lots of DNA inside their cytoplasm, but they don't have a nucleus. Whereas a eukaryotic organism has a nucleus. Animal cells have a nucleus. Plant cells have a nucleus. They're all considered eukaryotic. And if you think about the terms so biology is based a lot of biology is based off of Latin. Eukaryotic means true nucleus in Latin, and prokaryotic means pre-nucleus. So it just shows that one of the major differences is whether or not the organism has a nucleus or not. So microbiology isn't just about disease. We're going to focus mostly on disease, but it has such a wide scope that it's pretty incredible. In healthcare, agriculture, industry, environmental sciences, we rely on microbes extensively. This list of 3 things is nowhere near any the number of things we rely on microbes for. Food production, sure. Yeast and bread, right? You can have milk and add bacteria and you get yogurt. Making medications, penicillin is extracted from a mold sample. It's not anymore. We can now make synthetic penicillin, but it was before we could do that. It breaks down environmental hazards. There's something called bioremediation, where essentially you have microbes that help clean up toxic spills, for instance. So let's say the BP oil spill, right? There were microbes that were created to break down this environmental pollutant. So they added the microbe to the actual oil on top of the oil, and the microbes ate away at the oil, destroying it and breaking it down into something else, something much less toxic. So bioremediation is interesting. We don't spend very much time on bioremediation just because this semester we obviously have a shortened semester. One thing I do want to point out is that we really rely heavily on microbes in our gut to break down food in our digestive tract. So we swallow food, it gets to our small intestine, there's bacteria waiting, and then it breaks down the food, extracts the nutrients, lets us extract nutrients, and then the gut microbes can also synthesize some of the essential vitamins that we need. So we as humans can't synthesize a chunk of the amino acids that our body needs. We have to get it from our diet, or we also get it from the microbes in our gut that produce it. Agriculture is really interesting because in graduate school, one of my research focuses was agricultural microbiology, but we'll talk a little bit about that later on. So, as I said, pathogens are really are very less common than we think. We know about 1,400 of them, which is less than 2% of the known population of microbes that we're aware of. There are some pathogens that will always cause diseases in humans. So, for instance, Salmonella typhimurium, Shigella sonii, Bordetella pertussis. These names are specific bacteria that are pathogens, and their only job in life is to be a pathogen. Right? They don't help us in any way, shape, or form. They only harm us. So we'll talk about what we call a true pathogen later on. Now, an opportunistic pathogen is an organism that takes advantage of your weakened immune system or weakened state in order to actually cause illness. Now, a great example of this is on our skin. We have bacteria on and in our body everywhere. So let's say you cut your arm, your right arm, on a piece of rusty metal. What's going to happen is one of the organisms that lives on your skin, Pseudomonas, can actually enter the wound. So let's say it cuts your arm. Pseudomonas now has a way to enter the body through the cut. It will enter into the cut and it'll cause an infection, a skin infection. So the point being is when some organisms have the ability to be pathogenic, they can be. And we refer to those as opportunistic pathogens. Pseudomonas is a really, really good example of this. But it's not the only one. Another one would be Staphylococcus aureus. Depending upon the disease it causes, because it can cause many, it could be considered either a true pathogen or an opportunistic pathogen. So, point being, not all microbes are disease causing. We just happen to hear about the bad ones, right? No one ever talks about all the good ones that do amazing work to help us with numerous fields like medicine. They don't get credit. But now we know, I know, and now you know, that bacteria play a key role in all these fields, even though they don't get the credit they deserve, which is really unfortunate because bacteria that is used in medicine, I mean, it's essential. It's essential. Insulin, for instance, if you or anyone you know is a diabetic, and they have to use insulin, well, before insulin was produced on a mass scale, we'd have to harvest insulin from horses. It was a time-consuming and expensive process. Then we decided, hey, let's take the gene, the human gene for making insulin, and insert that gene into a yeast cell. And now the yeast cell, every time it copies its genome, makes insulin. So we started to produce insulin on a mass scale just by simply using a microbe. And this obviously was much more efficient. Much more efficient. Okay. So we'll cover some important scientists here. We'll go into more detail in some of them, but not all of them. So Hooke, he reported living things made of cells. Virchow, he added to what we call cell theory, where cells come from pre-existing cells. Leeuwenhoek was the first microscope and the first description of a microbe. Linnaeus is taxonomy, which is naming and classifying organisms. Pasteur proved biogenesis. We'll talk about what biogenesis is. And then Fleming was the scientist who accidentally discovered penicillin. A lot of people don't know that. He accidentally discovered penicillin. He was trying to grow a bacteria called bacteria Staphylococcus aureus on a plate, on a petri dish, and didn't work. Instead, he got a big contaminant of mold, and it turns out that mold produced antimicrobial properties that were killing the bacteria. And then we eventually, a few years later, had our first antibiotic. Very, very, very cool. Very cool. Okay. So when it comes to the test, you will possibly have to match the scientist with their major contribution. Now, if you're reading along in the book like you're supposed to be, you'll have noticed that there's literally double, if not triple, the names in the book. I do not expect you to know them. I only expect you to know the 6 that are listed here. I may add one or two more later in the lecture. I know I'll add at least one more, maybe 2, but these are the ones you want to focus in on. Oops. We need to talk about something called cell theory, because cell theory is kind of a segue into something called biogenesis. So cell theory is basically 3 tenets, 3 statements that we say about organisms, microbes, to define the fact that they are living, essentially. So the tenets of cell theory are the first one, all living organisms are composed of 1 or more cells. So the first part of cell theory is all living organisms are composed of 1 or more cells. The second one is the cell is the basic unit of life and can function independently, which is true. Think about bacteria. Single-celled, they can function independently. Good. And then the third one, which is what Virchow actually added to the cell theory. The other 2 scientists, you don't need to know their names. I'll tell you their names, but you don't really need to know those. It was Schleiden and Schwann, as well as Virchow, who developed cell theory. So the last tenet of cell theory is that cells come from pre-existing cells. Now, the reason I talk about cell theory before I get into the next slide is because Virchow's statement, or excuse me, Virchow saying that cells come from pre-existing cells was not always something that scientists and other people around the planet believed. There were a couple prevailing theories on how cells developed and how they worked, but and how they how they formed. We didn't have a good answer for that until a scientist came along named Louis Pasteur. We'll talk about Pasteur. So, spontaneous generation versus biogenesis. Talking about the origin of life on a micro scale. So we're talking about spontaneous generation where life, the thought is life can come from non-living materials like water, dirt, air, and they just automatically are created. Whereas biogenesis takes it in a different direction and says, okay, life begets life, meaning life creates new life. And cells have to come from pre-existing living cells. Now, if you think about that, if you remember meiosis or mitosis, let's say mitosis, you go from one cell to two cells that are genetically identical at the end of mitosis. So it even applies to human cells that undergo mitosis, that that life comes from pre-existing life. Our cells can't be copied and make new ones if they're not alive, right? So, key thing here for biogenesis, life has to come from living organisms. Now, there was a scientist, as I mentioned, named Louis Pasteur, who proved biogenesis to be correct over spontaneous generation. Scientists and theologists, as well as other people, argued for hundreds of years. I'm talking about 200 years, about which one was correct, biogenesis or spontaneous generation. And every time somebody would do an experiment, the other side of the coin, the other side of the argument, somebody would say, no, your experiment is flawed in this way. This is why it's wrong. It can't work this way because it's flawed. And they kept doing this back and forth for like 200 years until Pasteur came along and developed a single experiment that took all the previous experiments and built it into one. So it met every piece of criteria that they couldn't say, no, this is a fluke because you don't have this or this or this. No, he included everything, which allowed him to develop the fact that biogenesis is correct and spontaneous generation is incorrect. This is the experiment. Looks simple. and it kind of is. He took a flask and he put broth in it. Broth is a food source for microbes. So he took a flask, broth in it. Then in the first scenario, he left it alone. Notice the shape of the neck is in an S shape. We call that an S-shaped neck. Oops, sorry. And then in scenario 1, this media is sterilized. So he pours it in step 1. He sterilizes the media by heating it and making it boil, which kills any microbes that are in it. Right? If there's already microbes in the solution, then they were dead because they were boiled. Now, this sterile media was able to just kind of sit there and wait and see if something grew. Because if you think about the two topics, spontaneous generation, biogenesis, if it were spontaneous generation, then in theory, something should grow in here no matter what. Something should be given rise, because air can give, in spontaneous generation, the thought was air or dirt or water could give rise to microorganisms. But in this case, didn't happen. Scenario 2, he added the broth. He then heated these. He boiled them as well, just like the first one, to kill any microbes that were present at the beginning. And then what he did was he tilted the flask on this one a little bit so the broth went down partly into the S-shaped flask. And then in this one, he broke off the S portion and just had a vertical open tube. And in both situations, there was growth of microbes. Now, how do we break this down? Well, in our first scenario, nothing grew. But why? The question is, but why? Well, one of the major reasons is the fact that this S-shaped flask has two bends in it. So this bend right here and this bend right here. Microbes are heavy. Now, obviously, when I say something is heavy, the term is relative, right? Relative to their size. They're a lot heavier than viruses. So when it comes to trying to get to the broth, the bacteria from the air would travel into the tube and they get caught at the bend here or the bend there, and we're unable to get to the media, which is why it remains sterile. By tilting the media, in this case, the microbes only had to go a short distance within the tube to get to the media. And once you have one colony, one organism, it can copy itself really quickly to have a lot more present in that population. And then by breaking the S portion of the flask, having a vertical opening just allowed organisms from the air to enter into the flask. So Pasteur's experiment met all the previous experiments' expectations, and then they kind of agreed that, okay, biogenesis is correct, spontaneous generation is not. Germ theory of disease. So there are some standards on what we would consider a disease, how diseases are transmitted, among other things. Now, there's a scientist named Robert Koch who developed what we call Koch's postulates to determine if a specific organism causes a specific disease. So to say that again, Koch developed 4 postulates that help determine whether or not an organism, a specific organism, caused a specific disease. So he also developed some staining techniques, which was pretty cool. And then he actually studied Bacillus anthracis, which forms anthrax. Yeah, I don't think it was the best idea to work with that organism, but you know, sometimes scientists don't know better. Back then in the 1800s, mid 1800s, early 1900s, they didn't know anywhere as much as we do now, and they didn't have the internet to look it up, right? They couldn't Google it, so they learned the hard way in many cases. All right. So, there are 4 postulates. Now, in my syllabus, I mentioned that my PowerPoints are not complete, meaning you have to listen to the video and fill out information as you're listening. This is one of those times. It's a perfect example. I'm going to give you the 4 postulates and talk about each one of them, and it's your job to actually write them down. So first off, Koch's postulates. Now, if everything is met in Koch's postulates, we can say confidently that that organism causes that specific disease. So first is that the microorganism must be found, must be found in high amounts or high quantities in sick individuals, but not healthy individuals. So let me repeat myself. I had to cough, sorry. The microorganism must be found in high numbers in a sick individual and very low numbers in a healthy individual, if any. So if somebody's sick with a specific disease, they should have lots and lots and lots and lots of that pathogen. But if you were to compare that person to a healthy individual, chances are they probably don't have that pathogen at all. And if they do, it's significantly less, to the point where they don't have symptoms. Okay, so that's the first one. Second one, we isolate the organism from the diseased host and put it in a pure culture. So we isolate it. So we take, let's say, a blood sample and we isolate the bacteria, and then we go from there and we try to grow it in a pure culture. Now, a pure culture is when you have colonies on a plate, on a petri dish. Those colonies were actually derived from one cell. So if you can see it on a petri dish, there's billions of cells there. There's billions of cells. And we'll see images of that stuff later. So then once you have a pure culture of the pathogen that you think is causing the disease, you reintroduce the pure pathogen into a healthy host and see if you introduce it into a healthy host to see if that person gets sick. Now, I say person, obviously they don't do this on people. They use, in most cases, they use either primates or mice. So they will take that pure culture and they will reintroduce it into a healthy individual to see if that individual gets sick. Now, if that individual, that healthy individual, becomes sick from the pure culture that they were given, the last step is to pull samples from that organism and re-isolate the pathogen again. So you're re-isolating the pathogen a second time. And then this way we can try to identify from there what pathogen it actually is. So first one, microorganisms must be found, lots of them, in a sick individual, but not any or very few in a healthy individual. Microorganisms must be isolated from the diseased organism and grown in a pure culture. The cultured microorganisms should cause disease in a new healthy host. And then we isolate the bacteria from that new host. And we grow it and get we get pure cultures again. So these four postulates are really, really important. I would expect you to know them, because this is how we measure up to see if something's considered, pathogenic or causes a specific disease. Because one of the reasons we can meet these postulates. Now, not everything will fit in this, unfortunately. There are exceptions. For instance, there are some diseases that can only grow in humans. They're specifically human diseases, and they will not grow in a laboratory. They will not grow in other organisms. They will only grow in humans. So, we're not a country or, people where we just grab random strangers off the street and say, hey, I'm going to test you for this disease. I'm going to poke needles in you and take your blood. We just don't do that. It's unethical and it's dangerous. So it's really hard to figure out how those types of organisms fit into Koch's postulates when we can't really follow the necessary steps to prove those postulates. So there are limitations. That's a key thing. This is just an image in your book that shows you you have sick individuals that have a certain pathogen in it. We isolate the pathogen, grow pure cultures. We take pure cultures, We reintroduce it to a healthy individual, they become sick, and then we re-isolate the pathogen. We re-isolate the pathogen. Over here on the right, in that population, those organisms are not sick, so they don't have to worry about these other steps. So this is kind of what I was alluding to earlier. New diseases emerge often. Look at what we're dealing with right now. SARS-CoV-2, or COVID-19 as it's kind of generically called, is a novel viral disease. We've never seen it before anywhere on the planet. We just have never seen it, which is why we're struggling as a country, among other reasons. We're struggling to manage the spread of this disease, and part of that problem is the fact that we've never seen it before. Therefore, our bodies are not prepared to fight it. Therefore, we don't have a vaccine for it, and so on and so forth. So there's a lot of reasons as to why. But right now, COVID-19 is a perfect example of of a new disease that emerges. Microbes evolve new pathogenic capabilities. So this is interesting. There are many ways that microbes can pick up new traits, meaning they can share genetic material with each other, or they can pick up genetic material from the environment, or scientists can manipulate their genes in a lab. So they can evolve new pathogenic capabilities. And then think of what we were talking about earlier, how there are so many bacteria that we really don't know them all, but we know that only a small percentage can actually be cultured in a lab, which makes dealing with the other ones that can't be cultured in a lab that much more difficult. All right, this scientist, I do expect you to know, his name's Joseph Lister. Basically, he came up with something that was incredibly important, yet so simple that it was continually overlooked. He helped develop aseptic techniques, meaning sterile techniques. He also, when he did this, he was preventing nosocomial infections. So he was a doctor, an MD, and what he would do was he would use in surgeries chemicals to clean the wound, to clean the instruments, in order to sterilize everything to prevent infection. Nowadays, we don't even have to think twice about that. We know everything that they use to touch us is sterilized, in terms of surgical materials and pretty much anything. But back then, they would reuse needles. They They have a tool that they would use during surgery, and they just reuse it right after another surgery. So Lister finally decided to. He kind of was noticing a trend that kids in the children in the maternity ward were getting sick uh, quite often, and he wanted to know why. And what he found was that some of the providers, doctors and nurses, would go from surgery to the maternity ward to help deliver a baby. And maybe they changed their gown, their disposable gown, but it doesn't get rid of all the microbes. It's on your hands. It's on your face, possibly. It's all over you. Microbes are all over you from that surgery. So what was happening is these people were getting sick in the maternity ward because the doctors and nurses that were treating them were not sterile. They had microorganisms that they were transferring from the operating room to the maternity ward. And then this is where he really stepped up and said, okay, I'm going to try chemical sterilization. And he used bleach, or excuse me, not bleach. He used phenol. So phenol is a very volatile chemical. We don't even, as humans in a research lab, you should really never use phenol unless you absolutely have to, because it's an endocrine disruptor, meaning it messes with our hormone system. So back then, they had no clue. So he was cleaning instruments and cleaning the wounds with phenol. And unfortunately, I mean, phenol is a harsh chemical that could do a ton of damage to the patient as well. But it did sterilize the area. It sterilized the area. I've already talked about prokaryote versus eukaryote. The major difference is one has a nucleus, one does not. Hopefully you can tell me that prokaryotes don't have a nucleus, while eukaryotes do have a nucleus. There's some other traits that separate them apart, but we're really not going to worry about that. So morphology and physiology, basically for classification. Morphology is shape. Arrangement is how they group together. So anytime I ever ask a question that says, what's the morphology and arrangement of this image, I expect you to tell me the shape, this correct scientific shape, and then the correct scientific term for their arrangement. So here we have spherical cells, round cells. We call those cocci. Here we have single cells that are elongated. They're either rod-shaped bacteria or bacilli. Either one. The term is interchangeable. And then lastly, you have a spiral or spirochete bacteria where they are coiled. You can see that right here. These are the three major ones. Believe me when I tell you there are a lot of other ones. This just happens to be the three major ones. So you definitely need to know morphology and arrangements. So the three we talk about, cocci, bacilli, or rod-shaped, and then spirochetes, or spiral-shaped. We'll talk about a couple more as we get into diseases later in the semester. But for now, they're the three you need to know. And then for arrangements, if something is arranged in chains, we call it strepto. The prefix is strepto. If something is in clusters, the prefix is staphylo. And this image here shows good examples. Notice this top one right here. It's in chains. But also notice the shape. They're circles. They're cocci. So we can tell by morphology and arrangement that this organism is strepto because it's in chains, and it's cocci because the cells are spherical. Here in this second image down below, we have clusters. In microbiology, when we talk about clusters of organisms, we say staphylo, which refers to what we call grape-like clusters. And what's the shape? Well, the shape is they're cocci. So these are staphylococci. Staphylococci. Now the names you can see are giving you hints as to what the organism is looking like. Now you really can't tell right away without using a microscope and running some other tests to determine what the organism actually is, but you can gather a bunch of information before you run those tests so you have an idea of what if you have a good idea of what it could possibly be. There is one up here. You can see this is strepto. Chains for strepto should be 5 or more, so 5 cells or more. Sometimes we can get away with a little less, but should be 5 or more. If you have 2 of them, you have a diplococci. Diplo meaning 2, cocci being round cells. So right here, these are diplococci. Naming organisms. Oops. Linnaeus, he's the father of taxonomy, naming and classifying organisms. Now, there are quite a few rules associated with writing out organisms. So I'm going to tell you them right now. The first one is the genus is capitalized and the species is always lowercase, and you spell out both words in their entirety. So when I'm talking Staph aureus, what I'm trying to convey is I'm referring to Staphylococcus aureus. Okay. The correct way to write all this down is to have the correct full name, unless you're writing a paper and you use the name more than once. if you use the name more than once. So I'm writing a paper on Staphylococcus aureus. The first time I go to use the term Staphylococcus aureus, I write the whole thing out. But from there, you put a little parentheses next to that term, and you write capital S, period, lowercase aureus. You can abbreviate after the first time you've written it. The other thing is it has to be italicized. So should be italicized as well as fully written out. Now, what's the point, right? What's the point of having taxonomy and what we call a binomial nomenclature system? Well, for a really long time, scientists would work on the same organism and have no clue. You'd have a scientist in America working on, let's say, Staph aureus, and you'd have a scientist in Japan working on the exact same organism. And they would never know, because if they've never met, then you don't randomly just go up to somebody and say, hey, do you study bacteria? You know? I mean, if you do, that'd be interesting. But I don't. As a microbiologist, I've never gone up to somebody and be like, hey, do you study microbiology? That's a kind of a weird topic to start off with. So they never, back then there was no Google, there was no email, there was no Facebook, there was no whatever. So those scientists may have never met. But Linnaeus, by creating this binomial nomenclature system, made it really easy for people to say, oh wow, I'm working with the same organism as somebody else. They may be looking at different topics, but as long as they are aware that there's somebody else working on the same organism, there's no overlap or little overlap in terms of research, hopefully. We want to get solid research, but we don't want to repeat the same experiments 10 times. So it's important that we have this system in place. So Linnaeus's nomenclature system was really revolutionary at the time and allowed people to work on different or the same microbes and be able to communicate with each other and share this information, because they may have, in the past, before this system was in place, ever come in contact with one another, or even know that they were working on the same organisms. So moving on to the actual classification, our tree of life looks like this. It has 3 major domains: bacteria, archaea, and eukarya. And then from there, we actually get more and more specific. So you may have had a way of remembering it in high school, the taxonomic hierarchy. For me, I was always King Philip came over fearing green snakes. But back then, domains really were kind of a new thing. So now we just say, did King Philip come over for great spaghetti? And what that stands for is domain. So did is domain. King is kingdom. P is phylum. C is class. O is order. F is family. G is genus. And S is species. So King Philip came over. Did King Philip come over for Great Spaghetti? And then you take the first letter, and that's your taxonomic hierarchy. Domain being the broadest, and there are three of them: bacteria, archaea, and eukarya. Now, two of the three are eukaryotic, and you can tell by the names which two are prokaryotic. Sorry, two are prokaryotic. Only one is eukaryotic. So eukarya is eukaryotic, which means they have nuclei. Archaea and bacteria are both prokaryotic, meaning they do not have nuclei. Notice we have the common ancestor down here, and then there's divergence. What's really neat is a couple billion years ago, there were some bacteria that had the ability to generate energy by using the sunlight. And these bacteria were really efficient. And they eventually got incorporated into eukaryotic cells. So there was this inclusion of these organisms inside the cells, so they became part of that eukaryotic cell. Mitochondria is another example. Most of you probably hear constantly in science, mitochondria, the powerhouse of the cell, right? Our cells. The catch is mitochondria weren't always part of our cells. Okay, what happened is, or weren't always a part of eukaryotic cells. Let me make sure I make that distinction. Humans have always had, we can safely assume that they've always had mitochondria. But before that, eukaryotic organisms didn't have mitochondria. And then all of a sudden, a eukaryotic organism engulfs a bacteria that functions like a mitochondria, and instead of killing it and getting a little bit of energy, it just kind of leaves it alone, and it's like, well, go ahead, make tons of energy for me. And the eukaryotic cell took advantage of the fact that the prokaryotic cell made tons of energy, and it could take that energy away and just continue to do this. We call that the endosymbiont theory, that mitochondria and chloroplasts were once their own individual cells billions of years ago. Now, biggest question I get when I make that statement is, how do I know? How can I be sure that mitochondria used to be their own cells? There's actually some pretty important evidence that supports this. I'm going to talk about three important factors. Okay. First, if you look at our mitochondria and a bacterial cell, if you compare the two, there are three striking similarities. First, they both have a double membrane, an outer and an inner membrane. None of our other organelles have that, just our mitochondria, and bacteria do. Two, mitochondria have their own ribosomes, which are similar, they're more similar to bacterial ribosomes than they are to animal ribosomes. So size, structure, they're more similar to bacterial ribosomes than they are human ribosomes. Okay, so that's two really important ones. The third one is our mitochondria have their own DNA. They have their own circular DNA. Bacteria have circular DNA. None of our other organelles have their own DNA, but our mitochondria do. So based on these three, as well as other factors, we can say pretty confidently that mitochondria at one point in evolutionary history were individual cells that lived independent of eukaryotic cells. And again, when chloroplasts and mitochondria were incorporated into the eukaryotic branch here, we call that the endosymbiont theory. The endosymbiont theory. So here's another way of looking at it. Delightful King Philip came over for great spaghetti. Domain, kingdom, phylum, class, order, family, genus, species. As you go down the list, you get more and more and more specific. Right? Domain is the broadest. So let's say we're looking at eukaryotic organisms. The domain would be eukarya. The kingdom would be Animalia, which is animals. And then phylum, class, order, family, genus, species, you would have a specific list for each organism. And as you go down that list, it gets more and more specific. So you could have, for instance, chordata, or organisms that have a spinal cord, or mammalia, mammals. So you can go from really broad to really specific down by the genus and species. Hierarchy is something that's just useful for us. It's something we take advantage of because it allows us to say, okay, well, we're working on something similar. Let's try not to be redundant and try and study the same thing on the same organism, right? So anyway, hierarchy is very useful. I've already gone over domains. I'm not really gonna go back through that. Now, kingdoms, there are 6 of them. There are 6 of them. So you have domain and then kingdoms. Now, example for prokaryotes, prokaryotic organisms, whatever the domain is also the kingdom. So archaea, the kingdom, or archaea is also in the domain archaea. The kingdom bacteria is also in the domain bacteria. But fungi, plantae, animalia, and protists are all found under eukarya. And you can see that here, the domain they're under. But with bacteria, the kingdom is the same as the domain. So archaea, the kingdom archaea is within the domain archaea. Protists are really interesting. They're basically it's kind of I think of it always as a junk drawer. You have most everyone has one of those drawers in their house where you open it up and that's where you find a screwdriver, a pair of pliers, some Splenda, and other crazy random things. Uh, a TV remote. So the Kingdom Protista is kind of like that. It's a kingdom for organisms that are not characterized as plants, animals, or fungi. Um, and what's interesting is protists can't be lumped into one kingdom because they can be multicellular or unicellular. They could be um, autotrophs or heterotrophs, which autotrophs means it can make its own energy. Heterotrophs mean it has to consume food to get energy. So protists are hard to classify, and their kingdom is kind of the junk drawer of uh, of kingdoms. We just add stuff in there that we just aren't sure about, which is really funny. As a scientist, you think, oh, we really hope that we know everything, and we can really classify most things pretty well. Well, not so much. Sometimes you just gotta say, I don't know, and we can lump them together in our junk drawer. All right. So this is where I'm going to end on the first part of chapter 1. I will post chapter 1 video part 2 later at some point coming up soon. There's only about, I think, 15 more slides to cover, but you can expect. So the way I've built the course is we're following a Monday, Wednesday, Friday schedule as best we can. So I allot 2 class periods per chapter depending upon the chapter. So for instance, I allotted the first day of class, the 24th, and the 26th, Monday and Wednesday, for chapter 1. That's 100 minutes worth of lecture. Right now, I'm at an hour 17, so that is 77 minutes, okay, which means a roughly 25 minutes or so of finishing up the rest of this chapter. Sometimes I will go over that 100-minute mark, but there will be other times where I don't come close to that 100-minute mark. So it averages out to be about 100 minutes. Be aware of this, okay? So when you're watching videos, normally I don't break them into 2 parts. Normally I just have one long video. And when I do, you'll need to stop halfway. After 50 minutes, stop and then come back to it. Or after 30 minutes, stop and come back to it at some point. I don't recommend watching all of it at once, because by the end of the PowerPoint, you're kind of scratching your head saying, what did I just learn? Or what did I just read or hear? So, take breaks often and try not to go through everything all at once. Right? So this will be posted shortly, and then part 2 will be coming after that. Stay safe, and I will talk to you all soon.